I guess this will work now. Okay, I'm sorry now. I think we already now gained a few more participants, so very welcome all. Today we will host Dr. Giuseppe Suaria. He's joining us from Italy, I think. We will give you today in our webinar, this is the first webinar in the series of Microplastics for Breakfast webinars. I hope you have your coffee with me, I do. So we will have a presentation from Dr. Suaria, then we will have a short interview with your questions, then we will have live Q&A session where you will be able to live ask Dr. Giuseppe whatever you want. And after that, we will offer you our very own special feature right now, which is offering you a one-on-one discussion with Giuseppe after the end of this event. We had a very big number of people applying to this one-on-one session, and we were not really sure if we will be able to accommodate you all for this. We will try to meet your needs today, and hopefully everybody that applied will be able to speak with Giuseppe. So bear with us. We will let you know at the end of the event how you can stay with us, how you can mingle and wait until Giuseppe is ready to see you. Just to go through short housekeeping rules, please minimize your background noise with keeping your microphone muted. Please insert your whole name in your name part with the institution as well. You can post all your questions or comments in the chat section of the webinar, or you can use Slido. The link is the QR code is you can see here, and the number of the Slido. So please use that. If you have any technical difficulties, I hope not, please refresh your browser. This is something I was advised to do. Please, we will definitely appreciate your feedback at the end of this webinar. I will send you a feedback form in the email that I will send you after the webinar. So please reply to us what you need, what you liked. If you want to speak with Dr. Giuseppe in the chat, just raise your hand and we will make that possible. And this webinar is recorded, so if you will not be able to stay with us until the end, you can definitely get a link to the video YouTube channel where you will be able to see it later on. Now to tell you just a little bit about myself, who am I? I'm Andrea Palatinos. I was a microplastic researcher back in 2010 when European Marine Directive was taken into consideration, and we had to report the microplastics concentration in our marine waters. And since then, I've been working on microplastics more or less for a few years in laboratory, and later on through my experiences also in the commerce sector and at the ministerial level, I have been able to see the various aspects of this problematic. And then in 2019, I decided that I would really like to offer researchers a community, because in Slovenia, the research community grew immensely. So I organized first meetup of those researchers, and it was very nicely taken, and people want more. So I decided to do this community approach to join researchers from Slovenia, from Croatia, from all over Europe, and potentially also the globe. And Microplastics for Breakfast is a project of mine where I would like to offer you webinars and events with presentations of researchers' work and where you can communicate among each other. That's why I want to organize 4 of these events this year. The Giuseppe is the first one. Then in June, Dr. Francois Galgani will join me in a similar event. And later on, I have a plan to host a nanoplastic and the riverine litter researchers. So please stay tuned in order to join me also in the future events. And my supporter as well is Ljubljana University Incubator, to which I'm really grateful. They are hosting me as their mentorship. So, and they support me in my endeavors. So without further ado, let's move to Mr. Giuseppe Suaria. Just a short introduction about him. Here, Giuseppe, I took some information that are not so obvious. I know that you're from Italian, that you have a PhD in environmental science, but I didn't know that that your master was graded full 100%. So That's a very nice information. I also saw that you're an exceptional diver, that you took underwater photography, and that you're a BOSIET certified individual, which means that you are able to maybe save lives. So I'm not going to go into further. I just would like to maybe here focus or maybe give people a little bit information of what your researchers' focus have been. I would just simply say that you are a master of floating microplastic pollution, sampling and analysis, and that you have been working also on methods and standardizations, and that what drew me the most, that you were part of more than 30 expeditions. So I'm sure you have plenty of information to tell us. So I'm really happy that we have participants from all over the world. Giuseppe, I don't know if you knew that, but we have participants from US, from the South Africa, Brazil, Europe, Slovenia, Portugal, UK, Belgium, basically from many, many different places. A lot of Indians are also here today. So majority of the participants come from universities and research institutes, but we also have private companies and public institutions. So these are the people who are listening to you. Now I will stop my presentation and give you the floor.
All right, thank you, Andrea. Good morning, everyone. It's great to see all these participants from all over the world. Andrea, the introduction was too kind. Thanks a lot for that, and thanks a lot for organizing this. Can you please just quickly let me know if you see my screen?
Yes, we
okay.
see it.
Okay, great. So again, thank you for being here. I'm Giuseppe Suaria. I'm a research scientist at CNR. CNR is the Italian Research Council, is the biggest public research institute in Italy. And I work for the Institute of Marine Science, for the section of Lerici. So what I'm gonna briefly try to tell you today is a little bit of my story. How did I started to be involved in this field of research, and what are my main achievements in the last few years, and where are my research interests for the future? So where I think I will be going in the next few years. So I did started 10 years ago, almost 11 years ago. But when I started doing research, I was not interested in plastic pollution at all. I really was interested in these animals you see here. I was studying plankton and connectivity for a big European project, connectivity between marine protected areas in the Mediterranean Sea. So I was mainly interested in studying how the surface currents in the Mediterranean Sea will move these organisms from one place to another of the Mediterranean, and how these marine protected areas were connected to each other through the movement of water parcels. So, and to do this, I will go at sea. These are my very first oceanographic expedition I did in the Mediterranean Sea back in 2013. And I will be collecting samples of plankton from the surface of the Mediterranean. And I will use this net. The net you see in this picture here is a Newston net that will basically be collecting floating organism and plankton from the first few centimeters of seawater. And in this map here, you see the map, the distribution of all the samples I collected during those expeditions. But it was immediately clear to me while I was getting the samples and retrieving the net on board that the situation was quite different. So, and these are just some examples of some samples. And then you clearly see that the number of plastic fragments in these samples was much higher than the number of the organism I was interested in, I was supposed to be studying. So you see this sample here, I find it quite interesting. I don't know if you can read. We collected this in front of Naples. That says enjoy packaging. So the idea was made to start studying the issue in more detail. Several months of laboratory work followed after those samples to extract, count, characterize, and quantify all these tiny plastic particles. We in the end characterized more than 14,000 particles, and we published these results in a paper, and we called this the Mediterranean plastic soup. So where we described the soup of polymers floating in Mediterranean surface waters, more than 16 different polymers. with concentration of these microplastics all often very, very high. Some of the highest concentration ever recorded in the literature up to that moment. While collecting samples, I was also spending some times from the bow of the ship, like doing observation for macroliter, for floating macroplastics, so bigger plastic items floating nearby. In the end, I collected a lot of hours of observation for this paper. This paper was published in 2014, so about 10 years ago. And in this paper, we described the distribution of floating macroplastics in the northwestern Mediterranean Sea, estimating that there will be at that time about 62 million floating plastic items over the surface of the Mediterranean. This is an update because I kept doing this. It's my kind of meditation when I go at sea or when I join a research cruise. I often spend my free time looking the sea surface and recording all the items. And the update in these last 10 years includes now almost 10,000 liter items that I registered on my logbook and I recorded. So these data are not published yet. I'm working on it, and I hope they will be made available soon. And you see in this map here the current distribution of my observation around Italian seas. We are, of course, going forward from visual observations. Visual observation is very reliable, but it's not very efficient. So we are trying to develop, with collaboration with other colleagues, some automated identification methods using cameras and machine learning algorithms. And of course, the Europe a few years ago, the TGML came out with this master list for litter categories. So we want to harmonize the data reporting so that all these data can feed into database, which are then used by policymakers to draw meaningful policy to solve the issue. And of course, we can also use satellites. We have a couple of projects which are funded by the European Space Agency that are basically, we are testing whether the detection of marine litter from satellites is a feasible thing. It's doable, and we can probably say yes. We can use proxies. We can use this large aggregation of floating materials that you often see on the sea surface. We can spot these from satellites, and this will be a really breakthrough in marine litter detection and global mapping of the distribution of these plastic items. So, but back to the first few slides, that was 2013 when I was counting small plastic items or counting big plastic items from the ship. There were, as you can see from this graph here, just a handful of publications, just a few publications about the issue of microplastics in the scientific literature. Since then, since those first papers were published, the issue has really skyrocketed. You know more than me, I guess. Most of you are experts in plastic pollution research, so you know that the number of publications now is staggering, has been skyrocketing in the last few years. And this has been paralleled by the growth in plastic production, as you can see from the graph on the right. So I found myself in the middle of the cyclone, literally. And it was soon after, the first colleague started compiling all the available data about microplastic pollution in surface waters around the world. So, and this is the very first global compilation by Andres Cosar. And then there was, the same year, was another compilation was published by Marcus Ericsson. Then this very famous paper from Eric van Sebille came out, compiling again all the available data and nicely showing how the Mediterranean Sea was one of the most polluted regions of the entire planet in terms of number of particles, in terms of concentration of microplastics. Then two more recent updates came out in the last few years, one by Atsuhiko Isobe, by Professor Isobe in Japan in 2021, and one again by Marcus Ericsson in 2023, last year, that they updated with all the data that were collected in 10 years of research, their global estimates, and they unfortunately show a big increase in the estimate of plastic pollution on the surface of the sea. So, but if we do some maths, if we just take the numbers and we see all the available, like a few of the available estimates of plastic input from the global rivers, we see that this is normally in the range of the millions of tons of plastic per year. And these are 4 different estimates. which were nicely reviewed by Daniel Gonzalez last year. And if we compare these estimates of plastic input from rivers with the global estimates I just showed you about the amount of plastic floating at the sea surface, we see that there is a huge mismatch. So the estimates of plastics, of floating plastics, are in the order of tens, if not hundreds of thousands of tons. So there is order of magnitude difference between the two estimates. So then the question, of course, arises: where is all this plastic going once it arrives at sea? So of course, you're very familiar with this issue. This is basically the core question behind most of the research I do, and it is try to understand where is all the plastic going once it arrives at sea. This is another way of showing this. So we have this huge amount of plastic production. A fraction of this is mismanaged. A fraction of this will enter the sea, and then we only find between 1 and 3-4%, according to the most recent estimates, of this fraction entering the ocean on the surface. So this concept has been recently challenged by a few interesting papers, like the one I'm reporting here from Lisa Weiss, suggesting that most of this plastic is not really missing, but is just being stored in different reservoirs. For instance, the shoreline, the coastline around the world, that might be the biggest reservoir of plastic in the world. Most of this plastic is probably trapped in the near shore on the seafloor, so it's probably sinking as soon as it enters the marine environment. And so most of the bulk of the plastic budget is probably trapped in a process, in a near shore process, like of stranding and resuspension and sinking in the near shore. And this is again another way of showing like the core business of my research. So we want to put numbers, to replace question marks with numbers, basically, right? We want to close the budget exactly as we do with carbon or with all the other elements. We want to understand how much plastic is on the coastlines, how much is it in the water column, how much is in the biota, how much is on the seafloor. So basically trying to replace on a global scale these question marks with more precise numbers or estimates. And another thing that caught my attention when looking at this global compilation, this map is from the last paper from 2023. You see that there is a remarkable lack of data from the Southern Ocean, from Antarctica. And this is basically the main reason why in 2016 I embarked in this incredible expedition around Antarctica. This expedition was called ACE, Antarctic Circumnavigation Expedition. It was organized by the Swiss Polar Institute on board this beautiful Russian icebreaker. And I've been on this ship for 5 months, circumnavigating the Antarctic continent and stopping in about a dozen of different Antarctic and subantarctic islands, basically sampling for plastic and trying to put numbers to replace question marks with numbers. So we collected microplastic items on most of the beaches and the islands we visited. We collected sand samples for microplastics. We also did visual surveys and visual surveys for macroliter all around the expedition, all around Antarctica. And we only counted 54 liter items, right? So this is in more than 15,000 kilometers of observation. So this number is extremely low, especially if you compare to what I showed you before about the Mediterranean. The concentration are very low. And we also collected 33 samples for microplastics using another Newston net here. In these samples, we extracted 115 particles, but after analyzing them, we realized that 94%, most of these particles were actually paint chips from the ship or were non-plastic material. And so we were left with only 7 microplastics from all these samples around Antarctica, which gave us a concentration which is extremely low, is one of the lowest concentration ever reported from the world's ocean. And incredibly, north of 40 degrees south, so north of the Antarctic circumpolar current, north of the polar fronts, the concentration were 5 times higher if we compare them with some samples we collected in another expedition in the South Atlantic. And if we compare them, then I stayed on board the ship and I kept collecting samples on the way back of the ship from South Africa to Europe. And these data were finally published last year. You see them here. If we compare what we found in Antarctica and in the Southern Ocean with what we found in the Atlantic Ocean, where the concentration were about 10 times higher. And if we compare them to what I showed you before about the Mediterranean, well, the concentration in the Mediterranean are about 5,000 times higher than what we found in Antarctica. So what we concluded is that, and this was also clear while we were sailing through those waters, is that we were sailing in the cleanest ocean of the world. We were sailing in a very pristine seas, fortunately, until we realized this. This is one of the most exciting moment of my scientific career. We were on the ship, on the icebreaker, and there were some other colleagues sampling for phytoplankton, and they will be filtering water from a seawater intake on the ship, and they will be filtering this water on some very fine meshes, and they kept finding these colored fibers, the textile fibers in their samples. So they brought this to our attention back in 2016, and we were very curious. We thought these were plastic fibers, but we were not really sure from where these fibers were coming from. So one of the first one of the first hypotheses we did was that the seawater intake was too close to the gray water outlet. So we were sampling like fibers coming from our own laundry, from our own washing machines. And so to exclude these hypotheses, to reject these hypotheses, we went back to basics. We took a bucket and we went at the bow of the ship, in the front of the ship, to collect surface waters where the ship has not been passing yet. So, and we will be filtering these in the lab. And this is what we kept finding. Hundreds and hundreds of these tiny colored fibers from waters that were supposed to be pristine, right? Where the ship was not going through. And we were, keep in mind that we were in one of the most remote areas of the entire planet. So this was the unexpected finding, and we decided to add this sampling component to the rest of the expedition and to many other expeditions around the world. In the end, we collected almost 1,000 of these seawater samples from most of these areas you see on this map. We will be collecting replicate samples, and then we try to give a first estimate of the concentration of these fibers in surface waters. So contamination, if you work with fibers, contamination is a big issue. We will be washing everything with filtered water. We will be collecting aerial control samples to estimate the number of fibers coming from the air, from our own clothes, from the environment. We will be doing procedural blanks to estimate the contamination level during our sampling procedures, and then we corrected our data set according to the contamination levels. We then analyzed more than 2,100 of these fibers one by one to understand the composition. We analyzed them in the lab here in Lerici with our micro FTIR. We also did some other correction to our data set, correction mainly related to the mesh size. We saw that smaller, finer meshes will be collecting more fibers than larger meshes, of course. So we compute some correction factor. We also did some consideration about sampling depth. So because we realized that surface samples where the concentration were much higher than in subsurface samples. Also, we noticed that sampling volume had an effect on fiber concentration. So the higher the volume, the less the concentration. So probably because the fibers would be flushed down through the filters. And also we realized that replicates had a very low repeatability. So suggesting that single samples do not really provide a reliable idea of the realistic concentration levels in surface water. And we also published some recommendation and guidelines for sampling microfibers at sea. We published them a few years ago. So we provided this map. This is a first global estimates of the textile fiber concentration in surface waters around the world. These are some of the results. The concentration were varying a lot, up to 26 fibers per liter, but a median global concentration of 1-2 fibers per liter. The concentration of these fibers was not homogeneous across different basins. The highest concentration, again, as we saw for microplastics, were found in the Mediterranean Sea, as well as in Antarctica in the Southern Ocean. And the lowest concentration were surprisingly found in the Atlantic or in the Indian Ocean. There was a kind of a correlation of fiber concentration with latitude, and the concentration of these fibers was increasing going south, which is interesting because we are getting far from the human population epicenters of the world. We measure all these fibers for their dimension, their length and diameter. The length is about 1 millimeter, more or less, the median length, and the diameter is the typical diameter of the textile fibers we use for our clothing and textile materials. And there were also some differences in the size, in the length and diameter of the fibers across different oceanic basins, suggesting that the farther we go from the sources of these fibers, the smaller these fibers become. So the smaller fibers are those that are more easily resuspended and more easily travel around the world. But this is the most interesting result of that of this research. The most interesting result is that actually most of these fibers, the vast majority of these fibers, about 91.8% of these fibers were actually natural fibers of animal or plant origin. So most of these fibers are not microplastics. Only 8.3% of all the fibers we analyzed were synthetic polymers like polyester or nylon, while all the others were mainly cellulosic fibers like cotton or other cellulosics. There were also some differences again in the polymeric composition of these fibers across different oceans. And we can see that the number of synthetic fibers was increasing going south towards Antarctica, while it was lower in the Mediterranean Sea. And then we also tried very roughly, very brutally to compute, to give some estimates of the amount of fibers, how many fibers are floating around in the global ocean. And I was amazed to see the order in terms of number is staggering, is about 10 to the 18 fibers, floating fibers in the first meter of the water column. And in terms of weight, this is incredible because the total weight of these fibers in the first meter of the global ocean is in the same order of magnitude of floating plastic of the estimate of the global estimates I showed you before. So there will be the same amount of floating fibers as floating plastic particles. And then, of course, these fibers are very abundant. They are extremely abundant in the environment. They are everywhere, and they are, of course, interacting with the biota. They are being ingested by organisms and animals. This is the case all over the world, as well as in Antarctica. We collected fecal samples from king penguins in some of the islands we visited. And again, most of these fibers were, again, cellulosic fibers of natural origin, so cellulose, cotton, wool. And interesting, there were some differences, some significant differences between the reproductive state of the penguins, with incubating penguins being twice as contaminated as samples as chick-rearing birds. So it's interesting because these animals will change their feeding mode depending on their reproductive stage. And the issue is very important. So why I got so interested into textile fibers? Because if you dig into the literature, then this is a review from some of my colleagues that was published a couple of years ago. You immediately realize that if you dig into in the literature, you realize that most of the so-called microplastics, most of the particle counts found by microplastic studies around the world are actually fibers, with numbers are very high, higher than 80–90% of all the particle counts are generally fibers, textile fibers. So, but they are very different. In my opinion, they have very different sources, very different movements across the environment. And so I believe that they should be always considered as a very distinct class of contaminants, especially because most of these fibers are not made of plastic polymers at all. And consistently, this is being also demonstrated by other papers published in the last few years, that these cellulosic fibers, so these non-plastic fibers, are actually the most abundant fibers in many other compartments, not only in surface waters, in marine surface waters, but also in fresh water as well as in atmospheric fiber populations. So cellulosic fibers are very common. And if we classify them as microplastics without further analyzing them, we might overestimate the concentration of plastic. This is, for instance, an example from a paper that was published in Science a few years ago. Well, if you go in the supplementary material, you see that most of the particles that these authors counted and characterized as microplastics, they were actually fibers. More consistently, over 90% of these particles were fibers. So there is a very high probability that most of these fibers were actually cellulosic and they were not microplastic at all. And the issue of textiles, it's huge and it's growing exactly as the global plastic consumption. The global textile consumption is also growing. Um, and what is interesting is that the global production of textile material is different from the composition of the fibers we find in the environment, where synthetic fibers dominate right now the global textile production. They account for over 60% of the global production, but they only account for 8% of the fibers we find into the environment. So there is clearly something going on that really caught my attention, like why there is this mismatch, which is actually contrasting with the trend we observe for floating plastic, where polyethylene and polypropylene are the two most widely produced and consumed plastic polymers in the world, and they are also the most common polymers into the environment. But for fibers, the trend is not the same, is exactly the opposite, actually. And the causes for this are unknown. We are trying to understand, we have several hypotheses, we are trying to demonstrate or to further our understanding. For instance, one of these is that traditionally we've been considering that most of these fibers will be coming from washing machines. If you read the literature, you often always read about this washing machine as the biggest source of fibers. Well, it might not be the case, because just think about the number of textiles we use. They are everywhere, in cars, in tapestries, carpets, curtains, tablecloths, bed line and bed sheets, everywhere. Imagine a flag waving in the wind, or the dust you see during on your screens. Well, these fibers are everywhere, and there are some evidences from the medicine literatures, for instance, that most of the fibers are also inhaled by humans in very high amount. These pictures are taken from human lungs, and this is quite remarkable because 87% of the lungs contain microfibers, and 97% of the malignant lung cancer specimens, they had some fibers, cellulosic fibers in them. These are nice studies made by Francesca De Falco, my colleague. She was my colleague at CNR. Now she's in Plymouth in the UK. She nicely demonstrated that the release of fibers to the air just by wearing clothes, it's much higher and way more important than the release of fibers into wastewater when washing these garments. And she also nicely demonstrated that cotton garments do release many more fibers than synthetic garments. So basically challenging many of the assumptions that we had in the past. And also Imogen Napper, another researcher at the University of Plymouth in UK, she also nicely demonstrated, she compared the input of fibers through two different pathways, atmospheric deposition and wastewater from washing machines, and atmospheric deposition was way more important than the wastewater input of fibers to the environment. And that is why we are now starting studying the atmospheric deposition of fibers onto the Mediterranean Sea. So we want to understand how many fibers are depositing from the atmosphere on the surface of the Mediterranean, using our research infrastructures. And then also I'm also interested in what happens to these fibers once they arrive at sea, because for instance, this is a nice paper I found that was published in 2001, that showed that in a shipwreck, basically in a ship that sank in 1857, after 130 years under the water, a cotton waistcoat was still found there, intact. It has no clear sign of degradation after all this time into the marine environment. And this is the reason why I also started in the recent years to do some experiments using deep sea moorings to estimate the degradation rate into the deep sea of many common material, plastic, bioplastics, and textile materials in collaboration with Hydra Marine Science. And we are also interested in studying what's happening to the fibers once they enter into the water column. Most of these fibers, they are made of polymers which are heavier than seawater, so they would be sinking in the water column. And we are interested in studying, in sampling microfibers from the water column and trying to understand what happened and how they move around with in the water column, and also what is their sinking fluxes to the seafloor. So we are also using again deep sea moorings with sediment traps to collect the flow of these fibers to the bottom of the sea. We are doing this in the Arctic as well as in the Mediterranean Sea. We see from the first results, we see that there are some seasonal trends in the microplastic fluxes, which is probably related, is nicely correlated to the sediment flux as well as to the precipitation rates. So this is work in progress, and I'm really excited about these new results. And now I'm almost done. I hope I'm on time. And just a couple of slides on some other aspects, like not strict environmental research about the behavior, the movement of these fibers through environment, but also a few words for the bigger picture, right? This is why we do this. This paper, I found it really shocking, was published in Nature in 2020, and it showed that the global human-made mass now exceed the living biomass by far. So everything we build as humans is now much heavier than the total living biomass, which of course dominated by plant biomass. And if you go into the details of this, you see that plastic mass, the mass of all plastic ever produced by mankind, is about 8 gigatons right now, which is almost the double of the living biomass of all terrestrial and marine animals combined. And so these numbers are scary, are quite frightening, at least according to me. And this is the reason why we want to study this. And most of the solution we are developing are actually not really working so well. For instance, the many common biodegradable, so-called biodegradable polymers are not degrading at all into the marine environment because the sea is not a compost bin. Or for instance, it is often suggested to wear only clothes made by natural fibers, but these clothes, they release many more fibers than the synthetic ones. So is this really a solution? Yes, we are releasing less plastic, but we are releasing more cellulosic fibers, which are often dyed with chemicals and flame retardants and other chemicals. And this is my last slide, which is a bit ugly, but I find it extremely optimistic, which is about the patience and the publishing activity and the reason why we all do this, why we all spend years trying to publish our papers. And this is basically showing us that for many different contaminants in the history of humanity, these authors identified the onset of emergence, which is the yellow squares, and then they identified a peak in the scientific publishing activity, which is the red square, and then some regulatory actions, which are the black squares, and they found a common trend. So a common time period from the onset of emergence to the peak in publishing activity and to the onset of regulatory actions. And they forecasted that for microplastics, the peak in publishing, in scientific publishing, will be happening in 2022, plus or minus 4 years. So we might be exactly in the middle of the scientific publishing peak. We might be very close to the peak in scientific publishing. And so we hope that some effective regulatory actions will be happening soon. And all our hopes are on the United Nations Plastic Treaty. As you probably know, the negotiations are currently ongoing, and we do have our hopes there. So this was my presentation. Thanks a lot for the attention. I hope it was interesting to hear what I'm doing now, and happy to answer your questions. Thank you.
Thank you, Giuseppe, very, very much. I'm sure people are very interested in what you've now talked. And I would just ask you now whether our researchers that are now present here would like to contact you to be with you in some sort of discussion after this event. How would you say that this can be done, let's say, if they want to approach you maybe for some collaboration or whatever?
Yeah, I mean, just drop me an email, or and I'd be happy to answer anytime. You will find my email.
Can you just put it in chat maybe so that,
Let me check.
or yeah.
Yeah.
I mean, the whole thing about these events is to collect, to be like more in contact with each other. If you have any questions already, I would say please raise them now. That's a good time. We had one question in Slido, but we can now first focus on our short interview and then we will go to the questions. So my question to you, Giuseppe, would be: are you an optimist or are you a pessimist with regards to plastic? What would you say? I mean, the
Okay.
last slide said something about optimistic, but
Oh, that's a very nasty question. Thank you, Andrea. Yeah, no, I have to say that by nature I tend to be pessimistic.
no.
Extremely pessimistic. Um, yeah, I do not think, I mean, the problem of plastic pollution is strictly tied to the issue of overconsumption, overproduction, and our capitalistic world, basically, so, and our economies. So I am really afraid that the same failure we assist in regulated carbon dioxide emissions will be reflected into trying to regulate plastic pollution emissions. So yeah, I mean, by nature I am a pessimist, unfortunately, but it doesn't prevent me from keep doing what I do, because I think it's still useful somehow. And I keep the hope on. I try to keep the hope on, and to do my best for the sustainability issues and for the future of the environment.
What would you say is the biggest danger that we're facing with plastic pollution, in your opinion? What would be the most?
Well, in my opinion, plastic pollution is not and should not be our priority right now. It has become, because a lot of media attention and attention from the public and from the funding agencies was put onto this issue. But I personally think that if I was in charge of allocating funds and allocating money, I would redirect funding towards other issues like chemical pollution, for instance, or overfishing, which I think are way more pressing issues that humanity should address for the health of the environment. Regarding plastic, I do not see right now like huge risks for the health of the ecosystem, especially if we compare this issue with other issues we are having. So yeah, this is my answer, basically.
Yeah, there are emerging concerns now for the chemicals. I see that also in publications and discussions online. So that's something that, yeah, we're aiming towards, I think. You mentioned before that you're using also the satellite methodology to start monitoring or let's say evaluating the pollution. How was that? Is that a future, or what do you think? Is that something that researchers should be looking closely, or how was your experience with that?
Well,
Remote
it's
sensing.
yes, it's extremely promising. It's extremely promising. It's we are getting there. There are a lot of researchers, of remote sensing researchers working on this. European Space Agency is funding some research programs to try to understand the feasibility of this, and it's really promising, because if we are able to do this, and this can be only achieved though if NASA or the European Space Agency decide to implement a dedicated mission, so to deploy a dedicated satellite, a dedicated sensor on a satellite for plastic pollution. We already have the specification for that. The researchers know what they will need on a satellite to measure plastic from space. And if this is done, then yes, we can map the plastic concentration from space, and this would be extremely useful for many reasons. For instance, for mitigation, for targeted cleaning actions, for instance, or just for monitoring trends, just to see what is the trend, like is increasing, is decreasing, is our policy are being effective or not, what are the most impacted regions of the world, and so on. So I think it's extremely promising. It can be done. Of course, for macroplastic, we are talking about big items. For microplastics, I think it's pretty much impossible or very difficult.
Okay, I just cannot move on from the fact that you mentioned that funds should be allocated to other. Let's stay focused here today on plastic. Let's try to make people that do that work as much informed as possible. So in the future, how do you see microplastic research evolving? Which direction do you think the researchers should focus on or should be more aware of? What's the most important aspect, in your opinion, about microplastic that they should?
I think what is really urgent is to provide like a clear and solid scientific advice to policymakers about the impact of these particles. I have to say that by reading the scientific literature, it's not often extremely clear, that there are some contradicting evidences. And I guess if I have these kind of doubts, I guess these doubts are even higher if you go in industry, if you go to policy, right? So I think we really need to focus on providing a clear, scientific, undeniable, undisputable scientific evidence that these particles are having negative impacts on the health of humans and on the health of the marine environment, on the environment in general. So evidence is there, is growing a lot, but we really, I think, need to put a lot of focus rather than keep going at sea. Basically what I do, rather than keep going at sea and saying, I found plastic here, or here there are more than there, or this is sinking and this is moving there. I think we really need to focus, move in the lab and try to do experiments and try to understand what is the real toxicity, what with the real environmental concentration of these particles, with the real composition. We need to understand what are the real impacts on the organism.
What about you? What's your future endeavors in microplastics? Do you have already projects ongoing? Which ones? And do you plan to work more on microplastics? In which direction?
Yeah, I mean, I am in a whirlpool in a microplastic whirlpool. There are many projects, maybe too many projects, with dealing with different aspects, with the transport of plastic from to the Arctic, with the one I showed about the atmospheric deposition, about the degradation, about in the deep sea of these materials, about the effects. We have a new funded project on the effects of microfibers on the benthic ecosystem functioning. So as I say, we are trying to move our focus on the impacts of this particle rather than on the study of their movements and quantification. So I guess this will keep me busy for a few more years, although I'm also at the same time I'm slowly exploring other issues and other research fields. I feel that I need to do this after 10 years of research on plastic pollution. I'm also, this is on a personal level, I would like to explore different topics.
In Slovenia, we have a few companies, private companies. One of them is also here with us today, who are developing filtration solutions to prevent microplastics from entering into the human body or into the environment. What is your perspective on these solutions that are now being developed? It's mostly like filtering or coagulation or some sort of how would you maybe advise them to continue with their work?
No, I think it's very promising. I mean, I am familiar with Planet Care. I think Planet Care is a Slovenian company, right?
Yes,
I
as
think
well, yeah. Mm-hmm.
it's that is extremely efficient. It's also the only probably way of efficiently filtering fibers out from the washing machine. But as I said, as I also showed in my presentation, it looks like so this anecdote that the main source of fibers to the environment are washing machines is probably not true. If we Nobody has ever done the global calculations. We are missing these numbers. This has been anecdotically repeated in most of the scientific papers, but it looks like from the scientific evidence, it looks like that atmospheric deposition and release of fibers during normal use of the textile materials and not during washing, it's the most important source of fibers to the environment. And I think if I think about it also, it makes sense, right? And most of the fibers, if I just shake a carpet from my window, most of the fibers are released into the air, directly into the environment, not through the washing machines. Also because most of the washing machines, at least in the Western world, are connected to wastewater treatment plants, and which are actually somehow working to remove some of these fibers. Not as good as, for instance, Planet Care filters or other filters, but I think this everything is useful, right? But we should probably also start studying more the atmospheric release of these fibers directly into the air. If you think about, I don't know, beating a carpet or all the dust flying around us and depositing on our television screens, that is mainly made of tiny textile microfibers, and it's very difficult to get rid of that or to prevent shedding of those particles from our clothes. So, the issue is quite complicated. I think there are no easy solutions.
Recycling, yes or no?
Recycling, no. I mean, yes, now, but in the future, my ideal world in the future is a zero waste world, is not a recycling world. So we need to focus.
Reuse, repair.
Exactly.
Some sort of.
We need to focus on waste reduction, not on keep producing the same amount of waste and then trying to somehow recycle that. It doesn't make sense. We need to stop the production of the unnecessary stuff.
Yeah, everybody are with this the same approach. And one last question from me, and then we move to our participants. What would you say is your biggest accomplishment as a microplastic researcher? What are you most proud of in your 10 years?
Not my papers, for sure. I don't think my papers are of good quality, or at least they are not as good as those of my other excellent colleagues. I think what I'm more proud of is my reviewing activity. I don't know. I think it's very important, and I think now that I switched from being a reviewer to an editor for some journals, I understand what's behind the scientific publication process, right? And most of the time it's very difficult to find reviewers for papers. As an editor, it's extremely difficult to find reviewers. I have to send out tens and tens of review invitations just to get a couple of good reviews on a scientific paper. And the issue is really big. And but the reliability of what we do, the science we do, it's based on peer review, right? If we do not all commit to peer review, the whole system collapse, because we cannot guarantee the solidity of our findings, and we cannot go to a policymaker and say this is what the science is saying, because if everybody wants to publish but they don't want to review, the whole system collapse. So I think what I'm most proud of in the last years is the number of paper I reviewed and the effort I put into those reviews. And I think this was my main contribution to this field of research.
I think that's a good point here, as we have many researchers now present, to invite them to be active in reviewing processes as well. I know it's difficult. We mostly don't have time, but yeah, it's very important, as you said. Okay, I would move on now. I don't know if you can see my presentation. I'm sharing it. I hope you see it. Now
Yep.
I would open the floor. You are so very well behaved. Nobody's having microphone on. That's very nice. I would really kindly ask you if you can just raise a hand or post a question in the chat or in Slido. I will just post them now to Giuseppe that we got them. When I asked participants when they were registering to ask you question, we got one that is not answered yet. I mean, the one that what got you interested in the study of microplastic is already answered. You said that you were basically faced with the plastic instead of animals. And the second one was, can washing rice and soaking legume in water decrease the plastic content?
I don't know. We will need to do an experiment. I have no idea. So
Yeah.
I mean, I guess so. Yeah. I mean, washing, rinsing stuff, it's probably a good way of getting rid of some of these particles adhered on the surface of the items. But yeah, I mean, that then you will have to understand what happened with the water, with the rinsing water. But yeah, I don't know. It will not be efficient to remove the particles which are already incorporated into the organism and the cells in the plants. But yeah. I
Okay,
don't know.
let's go on.
Yep.
On Slido, there was a question, is there a procedure to remove pigments from pigmented plastics before Raman analysis to avoid peak interference?
Well, I've never been working with Raman, so I'm not really an expert on this, but I know that the pigment can be an issue. I saw a paper a few days ago actually that was showing how the UV will be degrading certain pigments, so and they would be responsible. The paper was studying how these pigments and coloration and dyes will be removed by UV rays. So, but not all of them. So some kind of dyes and pigments can be removed by UV aging, others not. So yeah, but again, I'm not an expert in Raman analysis. I mainly work with FTIR, so I don't know.
Whomever posted this question, if you are not still satisfied, just put your microphone on and let us know. So I hope we replied. The next one is from Mateja Grego from National Institute of Biology in Piran in Slovenia. What is the size frame of particles for remote sensing?
Well, the direct detection of single items right now has not been achieved. This is difficult. The resolution of most of the available satellites, it's too big to be able to see single items, single objects. What we are working on, what we found to be after a couple of 2-3 different projects, what we found to be the best way to achieve remote sensing detection of plastic litter is to use proxies. So, for instance, I briefly mentioned we are working with these windrows that are basically long accumulation of floating stuff at the surface of the sea. If you do sail, you probably sometimes saw these large, very long aggregation, which are often created by the wind, with the wind fields. We can detect these from satellites, and then through ground truthing studies, we can try to have an idea of what is the real concentration of this of the plastic items into this aggregation, into these structures, and then we can use them as a proxy for the presence of plastic and to get a number, a quantity of plastic, right? But single items right now, as far as I know, the detection of single items, unless we have access to military grade satellites, is currently not achievable with the civilian like standard sensors mounted on normal satellites. So yeah.
Okay. Mateja, does that replies your question? I hope she heard the reply. Then
Yeah, thank you. I heard it.
Oh.
Great, thanks.
Thanks.
Thank
You're
you.
also quiet. I'm a little worried. You can also put your cameras on so that we can see you. Thank you. So if that's a good reply to you, Matea, if you have any further questions, but I think you also have one-on-one with Giuseppe later. So let's move on to the next one. I don't know who that is, but it says, have you seen any animals that expressed interest in the floating litter? I wonder if they are just eaten incidentally and ignored, or are they causing some attraction and as a result, the animal behavior is changed?
Well, clearly, yes. I mean, I don't know about behavioral change, but the evidence it's now striking. It's clear. Many animals, many organisms are directly attracted, preying and ingesting plastic items. So, for instance, think about seabirds or marine turtles. All these animals are actually um, voluntarily, directly, deliberately ingesting plastic items. So they do mistake them for food. Some papers suggested that seabirds will be attracted by the smell of the algae growing on the plastic items. Anyway, these organisms they do directly ingest the items. So I guess this is already a behavioral change, right? They are ingesting something that it's actually not their normal food, not the food they evolved with for millennia and for thousands of years. So yes, they do directly interact with the plastic items. The marine turtles, for instance, they are one of the most affected. Almost all of the turtles now, they do contain plastic items in their stomach, as well as some seabird species. So yes, absolutely.
Whomever that was, I hope this replies to your question. If you have a follow-up, we're
Yes,
here for
hello.
you. Yeah, thank you, thank you.
Hello, it's me. My name is Mariana, and I'm at the UNG internship right now. My question is related to also my research, because I'm doing research on European tits and their behavior on the fibers, actually. And because the European tits are seem to be incorporating fibers into their nests, and I wondered that besides eating, do also like seabirds also they try to incorporate the plastics or the litter into their, I don't know, breeding behavior, or animals are doing like something else besides eating, or this is not noticed? Thank you.
Yeah, thanks for the question. Yes, I mean, again, I'm not extremely familiar with the interaction with biota, right? I mainly study the distribution and of these particles in the marine environment. But from the literature, from what I know, yes, there are several papers about the use of plastic, of litter, of different items, for instance, in the nests, in bird nests. This is widely published, and probably you are familiar with that literature. Some seabirds, they do also regurgitate plastic items together with the food items to their chicks when they feed them, when they go back to feed them. So yes, I mean, I absolutely, I should say, I think there are several kind of examples of these interactions with biota.
Yes, thank you. And one more question, because now I think about that, because you said that the fibers are usually, accumulated. So are animals like avoiding the places where the fibers are accumulated, or does it change anything, or is just like still like clear water and everything is swimming just like usual around them?
Well, these fibers are very small. They are few hundreds of micrometers down to few micrometers, 1 millimeter. So it's I'm not sure whether it depends on the organism, right? I guess we do not know whether the organism can sense them, can sense their presence into the environment, and then they will try to somehow avoid the high concentration areas. Actually, we do not have an answer to most of these questions, right? We really lacking a lot of studies about the impact of these fibers on the organism. What we know is that these fibers are everywhere. We are inhaling them, and so probably most of the other animals are inhaling them because they are flying around in the air. We are ingesting them probably because exactly for the same reason, because they are everywhere and their concentration are relatively high in the environment. So we are inhaling them, we are ingesting them, and probably most of the organisms are doing the same. But what we do not know is what are the impact of these fibers to us and to the organism. So as I was saying before, I think we really need to do more research on this. But to strictly answer your question, I don't know. I don't know whether the organism can avoid high concentration areas or not. It's a nice topic for further research.
Okay, that's all from me. Thank you very much for the answers.
Thank you. Thank you for the questions. Next is Maria Para. She also had her camera on, so thank you, Maria, for letting us see you. Thank you. Hi. She asked, what method of digestion of organic matter do you suggest for marine samples? Hydrogen peroxide, sodium hydroxide, etc., or maybe something else. What is the most efficient?
Well, we do work with KOH here with the Fenton reaction. I have to be honest here, I did not test it personally all of them. So to give you like a clear opinion, I should have tested all of them on the same matrix. And then I will be able to give you a clear opinion. We do work with KOH. It seems like it is the most, but simply because of the harmonization, because we also did some big European project about harmonization of methods and sampling methods and protocols, and we did systematic reviews, and we saw that the most widely used method was KOH. So in order to keep our results consistent with what has been done in the past, we choose to be consistent with the most commonly used methods. But of course, you know that it also highly depends on the matrix, right? So some matrices will be better treated with KOH, some other with hydrogen peroxide. So it really depends on what you are dealing with. If you are dealing with very nasty sludges or like organic rich sediments, you might want to use more aggressive methods. And so it really depends on what you're trying to do. I hope. Yep.
Satisfied, Maria? She's a researcher from Croatia, so yeah. Let's continue then to Zahur Manzoor. What's the minimum size of fibers we can analyze using ATR FTIR?
Okay, so with our machine, we can analyze. So the most important parameter here is the diameter of the fibers, not their length. Their length, it doesn't really matter. So, but we can easily analyze fibers, the most common textile fibers. The normal diameter of these fibers is between 10-20 micrometers. On average, like let's say 15-18 micron. So this is also our lower size limit for analysis for micro FTIR. So we can go down to 10-15 microns.
Okay, hope this replies to Zahur's question. Let's continue to Anna. Hi, Giuseppe. It was really lovely to hear your presentation. Very useful. Do you perhaps know if there were any studies on the additives and dyes in natural fibers found in the environment, or comparison of chemical burden between polymer and natural fibers?
Yes, hello Anna. It's nice to hear from you. I mean there must be a few paper, not that much honestly. We do clearly need much more research on these issues, because I think they are key. They are really critical. What I do know is that there are some projects starting trying to better study these issues, and I do agree with you. It's key to understand the impact of fibers. It's key to understand and why these fibers are not degrading in the environment. We really need to understand what what kind of additives and chemicals is the textile industry using to treat their materials, right? Problem is that most of these chemical formulation are basically secret. They are covered by commercial agreements. They cannot be they are not revealed by the companies. We do not really know much about all the chemicals and dyes and additives which are used by the textile industry. There are probably as many as the one used by the plastic industry. Yeah, there are, and also their analytical method to study them are not so easy. We need to use GCMS and this kind of methods, and there are not so many papers right now. But I do really hope that they will be increasing in the future. I'm not a chemist myself. I'm not an environmental chemist. I would love to be collaborating with some environmental chemists and trying to understand and to do something about the dyes and chemicals and additives, because yes, I do agree with you, Anna. It's really critical. This is a critical question.
Hi, Giuseppe. I finally managed to unmute.
Hi, Anna.
Hi. Yeah, it was a really lovely presentation, and it's nice to see you again and hear you again.
Thank you.
Yeah, and I do agree that definitely more experiments are needed to proceed with in this field. And it was really funny how you said that you're a pessimist.
I am. I'm sorry.
No, that's really cool that you're so honest. I am an optimist, but also kind of fearing that the future is not the brightest with the plastic, at least not the near future. Our near future is not the brightest with plastic pollution. So
Yep.
hopefully, I don't know, in like 5-10 years, it'll get better.
Finger crossed. Let's all keep our fingers crossed.
Yeah.
Yeah, I mean, Anna, I mean, just to keep on your question, I think what would be, for instance, extremely useful as a starting point is just some simple. If I was a chemist, just some simple leaching experiments, just trying to understand what is being leached by a common T-shirt into the seawater. That can be probably easily done in a chemical laboratory. Yeah, I don't know, but it's certainly this is something that we should be doing.
Yes, definitely. I mean, it is common that in plastic pollution research, chemicals are kind of not mentioned enough, not in focus as much as they should be, because it's basically they're intertwined, the polymer and the chemicals. So it would be good if they were studied together more than they are.
Absolutely.
And also what you said about the research, the reviewers, sorry, for the journals. Yeah, so I didn't know that it's that bad about with finding reviewers. That's also a really good call that you said.
It's extremely difficult. I started realizing this. I mean, I've been always reviewing because I always believed in this service to the community, and I also find it extremely useful for me to keep myself updated on what's going on. But as an editor, I really realized how difficult it is to find good reviewers. Most of the people will never answer your review request. Most of those who answer will be declining them. So it's like people are very inclined to be publishing, but very less inclined to be reviewing. But they do not. I guess we all need to understand that there is no publishing without reviewing. So
yeah. I mean, for me, the thing is that I normally decline if I know that I don't have enough quality time, to review it properly. I could review it, but I'm not, in a couple of days, but I'm not sure of the quality of my review. So it's basically all about the free time.
Yeah,
me,
I know.
at least.
I know. It's the same for everyone. Make no mistake, it's exactly the same for me, right? It's an issue, right? Because I think our reliability as scientists depends on the quality of the papers that are published and that they go through the peer review process, right?
Definitely.
if the quality of the peer review it's not enough, it's not high enough, then the quality of our statements it's low, right? And
Yes, definitely.
we lose strength basically. So
sorry, do I have another? Can I have another question?
Okay, I'll be quick.
Okay, no, I was just wondering if there are any interesting projects that are coming up open up for collaboration. Universities and institutions, Italy, Croatia, for example.
there must be some cooperation, like bilateral agreements. From time to time, those are activated by the ministries and they are published. Right now, I don't know of any specific cooperation program between these two countries.
Okay.
But cooperation can always be made. At least in my experience, it's very easy to start a collaborative project. Like we just start chatting. We just yeah, exactly. You just send an email and you just start working together, right? It's not. I mean, at least in my case, there's no problem.
Okay, but because there is at the University of Pula, they're basically starting their research into plastic pollution. So I was wondering, if there's anything happening. But cool, we'll definitely contact you.
I'm not aware of anything. Maybe Andrea is, but I don't know. I will let you know if I hear about something.
Okay, cool.
Yeah, Anna, that's a good question. I'm sure also others would be interested. So if we will hear anything, we will let you know through the community. So, yeah.
Awesome. Thank you so much, Giuseppe. And
Thank
Andrea.
You, Anna. Thank you.
Thank you. Hope to see you sometimes. Bye.
Bye. Let's move on to next question. Javier, I see you have raised hand, but we will first go through the question, if that's okay with you.
Yeah, that's completely fine. Sorry.
Yeah, Naira is waiting for some time.
I just wanted to comment on that. All the components that dyes and other chemicals that we can find in the. I don't know if I can just do a quick observation.
Yeah,
so.
Sure.
So yeah, it's just you're right, and sometimes the company won't share this information. Sometimes they don't even know. Okay, and yeah, because during my PhD we did some GCMS analysis of the water that they use for the first wash of the fabrics. Okay, so yeah, and we found from well, you name it, okay, even chlorine compounds, and it was like, wow, this is, and when we went back to them, they were like, okay, well, you will need to do a more further analysis, but we are not even aware that those compounds are released. So sometimes they are just like completely oblivious, to what they are putting out in the environment. And the problem also is that once you have found that there is a compound that could be potentially, damaging for the environment, for humans, for animals, and the time that you need to take to prove that compound is it's very bad, it might take a few years. By the time that compound is banned, it might take another few years, and in that time there is another new compound that it's practically the same compound but with another name or another brand, or they just done a small modification, and then you need to start the whole process again of analysis, verification, and then banning. So that's also one of the problems here. It's not that you find and you prove that there are toxic compounds. It's just all the time that it takes for it to be removed and how easily it can be replaced. So yeah, I think that's another problem that we have related with this issue of chemicals releasing from textiles.
Yeah, thank you, Avi. I fully agree with you. I mean, did you publish those results that you mentioned, like the GCMS study on the first wash?
We didn't, because that was at the end of my PhD. We just did the preliminary analysis, and yeah, unfortunately, I didn't have time to continue with them in order to get a full overview of them, because it will take like another year or two of my PhD, so it was impossible. I'm not sure if someone has taken after me, but yeah, it's something that definitely needs to be investigated a little bit more. I just realized that I was not even showing my face, so.
Yeah, no, I mean, let's try to publish these things, because as I said, I think I really agree with you. I think it's key, it's critical, and that's the wonderful world of chemicals, right? I mentioned this before. I think it's one of our priorities. We do need more research, and I also am perfectly aware that the regulation of these chemical substances is extremely complex. And I really think chemical pollution, it should be one of our priorities. So I fully hear what you're trying to say, and then it's yeah, absolutely. It's scary if you think about that you just then change something like something little in the formulation of the chemical compounds, and then you can start selling, you can keep selling actually the same substances, more or less. So
Yeah.
And this happens every day.
Yeah. In food and textiles and every aspect of the industry. So it's just, yeah, sometimes it's like a huge battle, against, something that I'm not sure we can even stop,
Yeah.
But well, we need to try.
I would love to see you communicate further, maybe change the contact, because we have questions waiting.
Yeah, no, I know, Andrea. Sorry for taking this time. Sorry. And thank you,
That's okay,
Giuseppe.
Javier. Lovely to hear you.
Thank you, Javier.
Okay, we have Naira, PhD student from Nova, University of Lisbon, and she has a question. I'm working on the quantity of microplastic ingested by marine species, and sometimes papers report only the number and size of particles. Do you recommend any paper with methods to convert the number and size to mass?
Yes, now it doesn't come on top of my mind, but I'm pretty sure that I've read something about this. There are a few papers that try to do conversion. Conversion of this kind were also made by some of those studies I showed you that provided like global budgets, global computation, global compilations. They do also face the same issue you are facing, so that some papers were only reporting the numbers, but they needed to do mass calculations, so they had converted from numbers to mass using some formulas. They are available in the literature. There are a few examples. So if you search, I'm sure you can find something. If not, just send me an email. I'd be happy to try to help you. But some researchers try to do something similar. Yes, absolutely.
Since there are many researchers now present here, I would also extend this question to all of you who are here. If anybody is aware of such a report or paper, just link it in the chat for Naira, or just DM her with this information. I'm sure she would appreciate it. Thank you. We have a reoccurring message from iPhone that he or she has a question, but I would rather go through the written ones first. This is from Vulezani Mayana. She's a student from the University of South Africa. My research is about some organic micropollutants and microplastic contamination in wetlands. I'm particularly interested in the impact of those on water quality, aiming to contribute to international allowed standards limits for water quality. Do you perhaps know of any development in microplastic standards for water quality?
Yes, you can check out the results of the EuroQ-Charm European project. One of the work packages was about producing standards, but I guess you are talking about water quality. Well, in this case, I know that there is some work ongoing, nothing really definitive yet. You can perhaps check also the Water Framework Directive from the European Union. Yeah, but and I also assume, yeah, Water quality, I guess you are not talking about drinking water quality, but like environmental quality. There are the baseline, contamination baseline established by the Marine Strategy Framework Directive, still a European directive. They establish like GES, like good environmental status of European waters. And so you can find some thresholds, levels there in the documents from the European Commission. But again, yeah, if you need more detailed information, I guess you can just drop me an email. I'll do my best.
I would also like to direct you to cost priority webpage where they have all the standards listed, and you can check them immediately. So I will just text you a link to that web page immediately when I will identify it. So if we continue on, I hope this replied to you, Vulezani. If we will send you the links in the chat. We have Anjali Tamrakar. What type of filter paper material you are all using to filter the samples? She basically put a question for all of the participants. So the type of filter paper material.
Yeah, we use GFD, so glass fiber. We find that it's the easiest to work with in FTIR, but there are many other options there.
Okay, I just found a web page with the standards. Okay, what Maria Paraj said, but we also replied to this one. Okay. So I think we now went through all the written questions, and now I would open the floor to the iPhone, who said that they have a question. Nobody's there.
Hello, I am from Bangladesh. My name is Rifat Jan Rakib. I am also working in marine litter and microplastic. I have some questions about detection of microplastic by using FTIR. How can I detect a polymer by using filter paper? And I have another question is that I want to collaborate with this guest author because he also a good scientist. I followed him in several social life sites like Twitter, LinkedIn, and many more. So I want to collaborate with him. Is it possible to collaborate with him?
Yes, hello. Thanks for the question. Yeah, absolutely. Just I'm always open to collaboration and scientific cooperation. So just send me an email. I'd be happy to discuss further. And yeah, regarding your questions, I'm not sure I really understood. Yeah, we do use a micro FTIR. So we use that to analyze the particles by Fourier transform spectroscopy.
But I want to say that how can I detect microplastic by using filter paper?
Excuse me, filters?
Filter paper, 0.45 micron filter paper.
Yeah, filter papers. Yeah, you can place them directly under the FTIR and analyze the single particles, and you will get a spectra of the particles. So that's what we do normally.
Just I give this filter paper under the FTIR, then I can easily detect the polymer, and I use the polymer library in the FTIR. Then it is very useful to me to detect the polymer.
Yes, exactly. This is exactly what we do.
Okay. And I also shared my email with you, and also I will give you email for further collaboration. That is my question.
Sure. Thank you very much. You can send me an email. There's no problem. I'd be happy to collaborate.
Okay. Thank you. Goodbye.
Thank you. Bye-bye.
Thank you so much. Our time now is right on schedule. We're 12 o'clock. We're now just about to finish the first part of the event and move to one-on-one sessions. Before we let everybody go, I would really like to now inform you that in June I will host Francois Galgani. I'm looking forward to him as well.